Hello everyone. Today we are going to talk about the mechanisms of immune evasion by tumor cells. From immunoediting concept, we already know that finally tumor escapes from immunosurveillance. During this process, many factors involved in immune evasion of tumor cells. Usually, we divide these factors into two aspects: intrinsic factors from tumor cells and factors from immune system. Here is the list for factors from tumor cells. We will look at them individually. As we mentioned before, tumor antigen is the initiator for anti-tumor immune response. However, in some tumors, there are too low amount of antigens to stimulate the immune response. Or in some conditions, the interaction of antibody with tumor cell surface antigens induce endocytosis and degradation of these antigens, which is called antigenic modulation. In some conditions, tumor antigens are blocked by antibodies, or the specific antibodies are blocked by soluble antigens, which would impede tumor eradication by humoral immunity. It has been reported that tumor cells always have low or deficient expression of MHC class I molecules, which leads to tumor evasion from surveillance of CD8 positive T cells. In addition, many tumor cells inhibit immune responses by secreting cytokines like TGF-beta and interleukin-2. They are well-known inhibitory cytokines and exert inhibition on many immune cells. Does anybody know exosome? They are small vessels, 20 to 100 nanometer in diameter, containing various DNA, RNA proteins, and expressing a lot of ligands on membrane. From this illustrator, we can see tumor exosomes play complicated roles in suppressing anti-tumor immune responses. The code inhibits the function of natural killer and T cells, repress the differentiation of myeloid or lymphoid progenitors, and promote the expansion, differentiation, and function of immunosuppressive cells. Additionally, tumor cells express death ligands, such as Fas ligand, to directly induce cell death of immune cells. Okay, these are intrinsic factors from tumor cells related with immune escape from immunosurveillance. Simultaneously, there are also factors from immune cells evolving in this process. Usually, we classify them into two aspects: the dysfunction of effector immune cells and the appearance of suppressive immune cells. T cells are the main executors in anti-tumor immune responses. However, in tumor microenvironment, some T cells become dysfunctional or exhausted. They are less proliferative, have low cytokine production, and express high level of inhibitory receptors like PD-1 and CTLA-4. Many factors involved in the development of exhausted T cells, including persistent antigen stimulation, hypoxia, low pH, and immunosuppressive cells like Treg, MDSC, and so on. Panel of inhibitory receptors are important for inducing and maintaining the dysfunction of T cells, such as PD-1, CTLA-4, TIM3, etc. Similar to T cells, tumor-infiltrated NK cells express high level of inhibitory receptors and low level of activating receptors. Dysfunctional NK cells showed low cytotoxic activity and cytokine production ability. Similarly, dendritic cells are also becoming suppressive, expressing inhibitory receptors and losing the ability to activate T cells, while inducing Treg production. Apart from the dysfunction of effector immune cells, there are a lot of suppressive immune cells in tumor microenvironment, like regulatory T cells, regulatory B cells, MDSC, etc. Let's look at these cells one by one. Regulatory T cells are the most well-studied inhibitory immune cells. In tumor microenvironment, many Treg cells are found due to trafficking, expanding, being differentiated from naive T cells, or converted from conventional T cells. Regulatory T cells suppress the activation and effector functions of other naive or effector T cells in contact-dependent or cytokine-mediated manner. Regulatory B cells are also found in tumor T cells. Unlike two common B cells, Breg cells have increased expression of membrane-bound TGF-beta, as well as PD-L1, which in turn suppress T cell proliferation, interferon gamma production, and inhibit CD8 positive cytolytic T cell activity, and then lead to enhanced tumor growth. Myeloid-derived suppressor cells, abbreviated as MDSC, is another type of inhibitory immune cell subset. MDSC could be classified into monocytic MDSC and granulocytic MDSC. The granulocytic subset of MDSC has high levels of reactive oxygen species, while the monocytic MDSC subset has inducible expression of nitric oxide synthesis. Then, MDSC play important roles in tumor development by either increasing angiogenesis, tumor cell stemness, metastasis, or by inhibiting the effector function of T cells, NK cells, and dendritic cells, or by inducing the production of Tregs. In tumor T cells, a huge amount of macrophages are infiltrated in. We call them tumor-associated macrophages. According to the activation mode, the phenotype, and the function, macrophages are classified into LPS or interferon gamma induced M1 macrophages and interleukin-4 or interleukin-13 induced M2 macrophages. Among of them, M2 macrophages are the main subsets of tumor-associated macrophages. Functionally, M1 macrophages enhance anti-tumor immune response and tumor destruction by secreting cytokines such as interleukin-2, while M2 macrophages promote tumor growth, metastasis, and angiogenesis by secreting TGF-beta or interleukin-10. Neutrophil, well known as an important player in acute inflammation, were also found in tumor tissues through the action of some chemokines such as interleukin-8. Tumor-associated neutrophils promote tumor cell proliferation, migration, angiogenesis, and inhibit anti-tumor immune response by producing cytokines ROS or MMP. In summary, in tumor microenvironment, effector immune cells become exhausted or dysfunctional, and inhibitory immune cells are accumulated, which then form suppressive immune microenvironment. That's all for today's lecture. Thank you for your listening.